Okay, so hello and welcome everyone. Great to see you here. I am very excited to announce today's speaker. Mia Jovanova is a PhD student at the Annenberg School of Communication at the University of Pennsylvania, and she does research on computational social science, neuroscience, network science, and all of that to study how people change their health behavior. And so her current work focuses on neural and social bases of the susceptibility to peer influence. And I'm very excited to have her here. As I said earlier, I'm very excited that here at the seminar we host speakers of all career stages. And Mia is our most junior speaker for this term so far, but that doesn't mean that I don't have high expectations for today's seminar. So join me in being excited, and I'll hand it over to Mia.
Awesome. Thank you, Alice, so much for the lovely introduction and as well for inviting me. I really, really appreciate it. And it's a great honor to be here and to be part of this amazing community. And so, as Ali mentioned, my name is Mia, and I'm a PhD student at the Annenberg School for Communication at the University of Pennsylvania. And today I will be talking about a project that asks how looking at brain network interactions can help us to better understand how people change their behavior in response to health messages that try to persuade them to be healthier and to change their actions in their daily lives. And this is interdisciplinary work that takes insight from network science and then applies it to other fields like health psychology and communication. And this work is really exciting to me because it shows the potential of how we can learn new things when we take insights from network science and then we apply them and extend them to new fields. And today I will be presenting work that is from a recent paper that was just accepted at Health Psychology Journal. Okay, and before I begin, I would like to thank my amazing collaborators across 3 labs at the University of Pennsylvania. So first, Emily Falk's Communication Neuroscience Lab, David Leiden-Staley's AHA Lab, and Danny Bassett's Complex Systems Lab. And I would also like to thank our funders for making this work possible. Okay, so to start, worldwide, the leading causes of non-communicable death and disease are largely preventable. And a large portion of these deaths and burden of disease could be prevented by better control and targeting behavioral risk factors like sedentary behavior, alcohol, and tobacco use. And today, in today's talk, I will particularly focus on sedentary behavior as a case study. So being sedentary is a really pressing public health problem, as more than a quarter of the population globally does not meet physical activity guidelines. And this is even worse here in the US, where more than 80% of the population is inactive. And to make it worse, recent meta-analysis suggests that inactivity has just become a bigger problem over the course of the pandemic. So when we take a step back and we try to think, okay, how can we change sedentary behavior? It is a really complex problem that requires both individual and structural approaches and strategies at different scales of analysis. But one important strategy at the individual level is health messaging. So typically, the health communication field has focused on developing mass media campaigns that encourage people to be more active. So here on the screen, we can see a message from a social media campaign that the WHO rolled out at the beginning of a pandemic to encourage people to be more active as they quarantine at home. Here's another example that reminds people to take a break from staring at their screens, something we may all relate to. But one recurring theme with messaging in general is that they don't always seem to work. Some messages may be more or less effective for some people to get them to move a little, but for others, not so much. And importantly, we see lots of individual differences from one person to another. So to go about developing more effective communication strategies, we need to understand the source of these individual differences and why are some individuals perhaps more receptive to messages than others. So how can we go about this? Well, one traditional way to go about it in the health communication world is we can show people messages. Then we can ask them to rate how effective they find these messages, as well as other constructs such as their attitudes, their beliefs, and their intentions to change their physical activity prior and after following message exposure. And then we can go ahead and link their survey responses to their future physical activity levels in their everyday life. So when we do this, on one side, we see there is some correlation between how people rate their messages that they see and their follow-up behavior. But there's also a large gap. So for example, I can rate a message as super highly effective, but then I may not go and act upon it in my daily life for many different reasons. So a complementary way we can also go about this question is that we can measure people's brain responses while they're viewing different messages in a scanner. And then we can take their brain responses and link them to their behavior outside the lab. And here, neuroimaging methods can complement survey responses because they allow us to see what actually happens in the human brain as message processing unfolds. And it allows us to capture processes that a lot of the time we may not even be aware of, and they're not captured by self-report. So here what's really interesting to me is that people's brain responses in the scanner are usually not correlated with their self-report and how they feel about the messages, suggesting that we perhaps may be capturing different sources of variance when we try to explain behavior. So when we think about how people evaluate messages, a common theoretical framework from cognitive science and health psychology suggests and draws on these models that are called value-based decision-making and expectancy value theories. And briefly, these models argue that people make evaluations towards messages by making value computations, and they do this without much cognitive effort. So here, the brain integrates different decision inputs and weights them on the same scale via a common neural currency. And one consistent finding in the literature is that there are a set of brain regions that track with these subjective value signals that we believe integrate different signals from other parts of the brain and summarize this in an integrative summary value signal. And a large body of work suggests that individuals who show stronger activity in these brain regions associated with tracking subjective value signals in response to messages may also be more receptive or also more likely to change their behavior in response to these health messages. And further, so research suggests that messages that are perceived to be more salient and more self-relevant are also more likely to be encoded as subjectively valuable, and in turn to elicit greater behavior change. So based on all this work and theory, we can consider salience and self-relevance as two inputs that feed into subjective value computations in the brain, and to be particularly important in how people respond to messages. So based on this framework, in today's talk, we're going to ask, how does the brain integrate information about what people can find self-relevant and salient? And to do this, today we're going to focus on two functional brain systems in particular. So first, we have a set of brain regions, sometimes referred to as the salient system. And these regions are often implicated in attention, emotional processing, memory. So for example, we draw on these regions when we look at a health message in a very busy, noisy environment, and they may help us to detect it as salient, or this is important. Next, we have another set of brain regions that are sometimes referred to as the default mode system, which support what we often do by default, which is very often think about ourselves, our values, our preferences, or about others. And a large body of work suggests that there are specific regions that are part of the default mode system that track with greater subjective value. So traditionally, most of the work so far within this health, social neuroscience domain that is focused on messaging in particular has focused on just the role on individual brain regions and within these systems, and has found that these isolated brain regions are associated with greater message effectiveness or greater message receptivity. But what is interesting is that these set of regions are very densely connected within their own functional system, in particular to support specialized function. But they also communicate with one another. So there's a lot of between system crosstalk, as well as with additional brain systems that are beyond the scope of today's talk. So here we're extending this line of work to ask, how do these two brain systems work together versus alone during message processing? And one possibility is that greater crosstalk between the two systems may allow the default mode system to integrate inputs from the salient system, like attention, saliency, and affective responses, into a greater summary signal of subjective value towards the health message. And in turn, these processes can facilitate message receptivity. And here comes the really fun part. So where to approach this question, we can take tools from network science and we can conceptualize the brain as a network. And to do this, we can first extract time courses while people are viewing different health messages. And next, we can construct a network where we can consider the brain as an undirected weighted graph, where we can consider brain regions from these two systems within this context as nodes. And we can consider the temporal correlations in activity between regions, which tell us how closely activity in these regions fluctuates together as edges. We can quantify the strength of connectivity patterns both within each of these two systems, as well as between the two systems. And on one side, communication both within and between is super important to support information processing. But what's really interesting is that these connectivity patterns vary a lot from one individual to another and across different contexts. So based on existing work in the functional connectivity domain, we believe that studying individual differences in connectivity patterns both within and between the default mode and the salience system may be particularly important to understand health message evaluation. So in this paper, what we do is we focus on the relative balance in the between to within between connectivity strengths. More specifically, we ask how does the extent of the default mode and salience network integration meeting the relative strength of between to within network connectivity, specifically while people are viewing health messages, relate to their future changes in physical activity. And to test this as part of a larger parent study, which was led by Yuna Kang and is now in PNAS, we recruited 150 adults who reported very low physical activity levels and high BMI. First, participants wore wrist trackers during the day for a week with the goal to just measure their baseline levels of activity, just how regularly they move in their day-to-day life. Next, we had 150 participants undergo an fMRI scan, where they viewed around 30 different health messages that encouraged their physical activity and they discouraged being sedentary. So here's one example of the stimuli. When you notice that you've been sitting for a few hours, stand up. And here's another example. Try to stand up, stretch, and move around as often as you can. And now these messages are on purpose plain to avoid confounds with any additional message characteristics. So here we just try to focus to minimize any other features, and we just try to make them as plain as possible. And participants also viewed a control set of messages that were unrelated to physical activity, and I'm happy to chat in more detail about the message development process in the Q&A. So after the scan, we tracked activity levels for 4 weeks as participants went about their lives. And participants also received text messages that reinforced the health stimuli they saw in the scanner. So we were reinforcing this messaging intervention for the course of the month. And we were really interested in capturing behavior change. And what this means is how did people's activity levels change in the post-scan period and while they were receiving these text messages while to prior the scan. And we operationalized behavior change as the difference in the post-activity messaging period versus the week prior to the intervention. And following standards in the physical activity domain, we further broke down behavior into 3 types of behavior change. And the first is just reduction in time spent sedentary, that people reduce the time they spend not moving or sitting. Next, we looked at light activity, which includes activities like slow walking. And finally, we also looked at moderate to vigorous activity like jogging, which is perhaps a lot more difficult to change. And on the brain side, we were interested in the crosstalk or the integration between the default mode and the salience systems during messaging. And now I'll briefly go over how we calculated this measure. And the figure I'm showing you here is a correlation matrix between brain regions that span the salience and the default mode, and it's averaged across all individuals. For each person, we first calculated their average connectivity strengths of regions that span just the default mode, as boxed in blue. And then we calculated the average within connectivity strength within the salience system, as indexed in blue. And then we just took the average within connectivity from the two systems. Next, for each person, we also calculated the between connectivity strength between the two systems of interest. And finally, similar to prior work, we derived this measure by subtracting the within from the between connectivity strengths. So what does this measure actually tell us? Well, we see relatively stronger integration when the connectivity between the two brain systems is strong relative to the connectivity within each one on its own. So next, we took and we used this measure as a message receptivity index during message exposure, and we correlated it with future behavior change. And specifically, we hypothesized that individuals who show greater integration during message processing, meaning they will be perhaps more likely to integrate salient and self-related processing, may be also more receptive to the messages or more likely to change their behavior inconsistently with the themes from the messages. And we also looked at these three behavioral outcomes separately, as these can have related but often different effects on health, and as it's commonly done in the physical activity domain. And now let's show you some of our results. So on the x-axis, you will see brain integration scores, and on the y-axis, you will see behavior outcomes. And first, we'll look at changes in sedentary behavior. So we found that greater integration was associated with a decrease in sedentary behavior, such that people who showed greater integration to the messages were also less sedentary in the post-scan month. Next, we looked at changes in light physical activity. And consistently, what we see here is that individuals with greater integration were more likely to increase their light physical activity during the post-scan period. And I should also mention that in all the models, we controlled for variables such as demographics, gender, age, education level. And finally, we found no relationship between brain integration and moderate to vigorous physical activity, such as running or jogging. And there are several possibilities that may explain this null result. So first, the sample in this context was very sedentary, so this is perhaps the most difficult outcome to change. And in addition, there was the least amount of variance observed in this measure. Also, if we go back to the actual example message stimuli, it is interesting that many of them targeted lighter activities, but the language itself didn't necessarily target more moderate activities. So, for example, there was very little mention of trying to get people to run. So together, the results we have so far suggest to us that individuals who show greater integration during message processing were more likely to act in message consistent ways, such as to decrease the time spent sedentary and to increase their light physical activity over the course of a month. So next, we conducted a number of follow-up analysis, which I will go over briefly. So first we asked, what are the default mode and salience system integration relates to behavior change when we control for each person's just within connectivity strength and between connectivity strengths separately? And what we found that yes, which suggests to us that it's the relative relationship between the two that is really driving the results versus just looking at each one on its own. And next, we asked, does the default mode and salience system integration relate for behavior change above and beyond controlling for self-report measures like attitudes, norms, efficacy, and intentions to change people's physical activity levels? And what we found that yes, which suggests to us that what we see in the brain is capturing different sources of information or different processes that what we can capture is with self-report measures within this specific context. And finally, we asked whether the findings that we see are specific to these two systems of interest, which is the default one and the salience. And to do this, we shuffled or permuted the nodes that belong to each of these networks, and we found no results when assigning nodes to systems randomly, which suggests some support for the brain system specificity within this given system. Of course, there are other relevant systems that also contribute inputs in the given context, but we have not explored them or looked at those yet. So overall, it is possible that individuals who show greater integration may be able to more effectively integrate salient and self-related information processing while they're viewing messages. And in turn, they could be in a more optimal brain network state to attend to and to act on these messages when they go out and make decisions in their day-to-day lives. So this work in particular highlights the integration of brain systems that process saliency and self-relevancy in the context when we want to use messages as a tools to promote behavior change. Now, one question is, okay, this is great, but what can we do with these findings? And here I will outline one approach. So in the next steps, we can go back and look at a wide range of different health message stimuli. What we can do is we can ask which of these message types are most likely to elicit the greatest default mode and salience integration scores. And we can do this in a subset of brains using neural focus groups. So by using this, we can ask which are the best performing messages at the neural level. And then we can take these messages and also test the real world effectiveness in an out of sample contest. So here we can ask, do the messages that evoke the highest integration scores in the brain are also the ones that are most likely to perform the best in the outside world and outside the lab? And what this approach does is it allows us to take what we learned about brain integration during message processing, and we can expand it to ask how reliably can brain integration provide a marker of message effectiveness across different behavioral contexts and also across different populations. So finally, to sum up, we found that looking at brain network interactions between the default mode and the salient systems can help us understand who may be more or less receptive to messages that promote physical activity. And to contextualize this work, it highlights the value of expanding the focus from just looking at individual brain regions in the brain to just broader and larger scale system connectivity. And the implication of this line of work are twofold. So on the brain side, we're learning more about what actually is going on and what happens in the brain during information processing. And then on the flip side, on the practical side, we're trying to develop new strategies to improve health and well-being. And that is all I have. And thank you so much for listening. And I'm super excited to take questions. And I hope this was okay, Alice, in terms of your expectations. I took a bit shorter than the full period.
This was awesome. Thank you. I think usually people don't mind when talks are shorter than they expect. So as always, we have time for questions. So if you have a question, you can type it in the chat, or you can raise your hand, and I'll try to see who's in the chat. So to get us all started, one thing that I think I just missed was something about comparing. So the measure that you used, integration, that's essentially comparing the connectivity within your clusters, your functional groups versus between the functional groups, right?
Yeah.
And so that I think you mentioned something about comparing that to some sort of baseline per person or something, because I assume that even if you don't show people any health messages, you would see differences between people, right?
Yeah, that's a really, really good point. So you're tapping onto something here that's like super important, is these functional connectivity individual differences, to what extent do they stem from the message itself versus just like person to person differences? So one way we try to approach this is we compared functional connectivity while people are seeing the messages that are related to physical activity, but then also when they're seeing messages unrelated to physical activity. So that is kind of our control group, which is what happens in your brain when you're looking at messages that are about the environment or climate change. And then we looked at functional connectivity patterns there. And what we found is that those functional connectivity patterns are not associated with behavior change, but the functional connectivity patterns during message exposure, while people are viewing physical activity messages, are associated with the specific behavior change that we're targeting, which to us suggests that it could be something that is happening that's specific to the messages themselves. So I think this needs a lot more work to be done. This is like a very early stage open area of research. But yes, does that somewhat answer the question?
Yeah, definitely.
Okay.
Yeah, that's great. So we have a question in the chat, and we have a raised hand. Let's go with the raised hand first. So that's Jen. Do you want to go ahead and unmute?
Hi, yeah, can you hear me?
Yes.
Hi, thank you so much for a really lovely presentation. It was really fantastic and really interesting work. I'm Jen Greaser. I'm actually a lecturer at the University of Exeter in the UK, and I look at brain networks, and I've looked at functional connectivity from fMRI as well. And one of the things that has become more popular recently is to, instead of looking at the average functional connectivity, is to look at how that varies over time. So I assume you're showing this picture for a certain amount of time. And so how does that functional connectivity vary during that time? Is that something that you've looked at? And then kind of maybe a related or slightly different question is in terms of the networks and the network structure itself. So there are lots of sort of graph analysis tools that you can use to kind of tease out some of the important connections. The default mode network you showed is really big. For example, that seems to be loads of different regions. So are you able to identify any specific regions that might indicate causal mechanisms for this additional receptiveness?
Great question. First, thank you so much for your kind comment on the talk, and I'm super excited that you're working on functional connectivity methods. So the first question is about dynamic functional connectivity. So we have not done that yet because the way this task was designed, it was a bit complicated to isolate the time windows in order to do that approach, but we're very excited to do that in next steps, particularly because then we can look at measures such as flexibility. So we can look at how nodes change connections over time. And there is some evidence to suggest that greater flexibility, particularly in the ventromedial prefrontal cortex, which is part of the default mode, should be tracking subjective value signals and integrating these different signals over time. And it should be associated with greater message receptivity. So I think that is a great open new area to go towards there next. In addition to that, for the second question is, yes, the default mode network is really large, especially relative to the salience. There's a lot more nodes. So one way to go about this and to ask what are the components of these systems that are driving the effects is that we're thinking of doing community detection on the default mode system. So trying to parse this in a data-driven way. And then based on this, try to see what is it that is actually driving these results. And so far, the preliminary evidence we have is that it's all driven by the ventromedial prefrontal cortex. So when you isolate it, the components, that shows up as a cluster, and that seems to be most strongly integrated with parts of the salience systems.
That's great. Thank you very much. That's really interesting stuff.
Yeah, so I see a comment in the chat. Lorena's asking whether you can disable the screen share so that we can all see each other. Lorena, thank you so much for the feedback. And we have another question from Francisca. Do you want me to read it out, or do you want to ask yourself?
Can do it. So I was wondering if you have any future plans, or if you are considering maybe another follow-up with the people or another scan in the future or something like that. If you can maybe tell us about that. Thanks.
Yeah, so I'm interested in thinking about generalizability. So for example, this project is really cool because we have a big sample, but it's just in the context of physical activity. So what I really want to know, is this something that's just specific to physical activity, or can we translate this to other contexts, like for example, alcohol use, because other behavioral contexts, because I think it would be cool if these results replicate across different contexts. So right now I'm working on a project with Alice and Peter, which is focused on alcohol use. So I'm hoping to take some of the methods that we developed here and try to apply them in a context to see how people respond to stimuli in response to drinking. So that is a part where I'm hoping to take this work next. Oh, I think you're muted.
Okay, so thank you, Francisca. So if I may, I have another question that I wanted to ask you, which is, you basically had these 3 different types of behavior change, like just being less sedentary, low activity, high activity kind of thing, and you didn't see any effect on the high intensity activity as you saw for the other 2 categories. So one thing that I was wondering about is whether this is kind of something that is very based just on the fact that finding time to go for a jog is something very difficult, and so it's very difficult to nudge people into that, or whether you would think that with just a different type of messaging or looking at a different brain region, you might get results where you get a positive correlation for that.
Yeah, it's a great question. So I think there are 2 pieces to it. So on the first side is that I think it perhaps has to do with some of it with the sample itself, where we particularly focus individuals who are sedentary. So it could be that this is the most difficult outcome to change. In addition to that, some of the variants could be related to just the content of the messages. So it could be, I think what this work did not do is, what we could do next is that we can experimentally assign individuals to different message conditions. And then if we can try to isolate to what extent is it message driven versus individual driven. Now, one thing, and this is not specific to the moderate vigorous activity outcome in particular, but more broadly, I think something that is super complex here is that just physical activity is such a complex and multi-level behavior. So for example, I think that the messages can only do so much. So for example, if it's, let's say, a very low SES setting, if there aren't safe environments to work out, I think there can be a lot of other factors that are competing and going on at the same time. So with the messages, we're like targeting one aspect of it, but then again, I think we need a lot of structural level interventions as well.
That is super interesting, and to be honest, that the last part that you said actually reflects some of my own personal experiences, where during the pandemic, when everybody was working from home, I went to see a physiotherapist, and a person said to me, you're not walking enough, that's why your ankles hurt, you need to start walking every day, go for a walk half an hour at the least. But I was living in a very densely populated neighborhood with no common like green spaces around, so like my options to go for a walk were like basically walk along the highway, and so I never did that until I moved somewhere else where there was actually like a nice park where you could go for a walk.
Yeah, which is a really big, I believe, when it comes to the messaging part, I don't think that negates like, oh, we shouldn't have messages because at the end of the day it's all like environmental, but we can ask, okay, what are people's actual circumstances? And we can try to tailor and develop messages that are most likely to be effective within that given context. So for example, let's say if I don't have any spaces to go outside, maybe a message that reminds me to do something different may be more effective to me, and it won't be effective if it just tells me go walk outside where I actually can. So yeah, thanks for bringing that up. That is I think in the practical public health applied setting, that is kind of a really, really big, important open question now. It's like, how do we design more effective messages?
One thing might be community driven, and that's something that we're working on right now, right? So there's a few more lovely comments in the chat about your work. If there are no further questions at this point, I'm going to close the session. And thank you all for being here. And thanks again, Mia, for this wonderful talk.
No, thank you so much. I really appreciate it. I'm very thankful for you